Hi everyone. I welcome you all to the another lecture of this series on metabolism. So today we will be learning about the absorption of carbohydrates. So see, we so far we have covered the digestion of the carbohydrates, and now from the intestine the carbohydrates they will be absorbed absorbed to the blood so that they can enter to they can go to the cells and there they can synthesize the ATP. Okay, so when we talk about carbohydrates, they are absorbed as monosaccharide from the lumen of the intestine, and so far as uh, co- concerned, so far as we've concerned, there are two mechanisms by which the carbohydrates they will be transported into the portal circulation. Okay, so first of all is the active transport. In the active transport, the carbohydrates, let's see the monosaccharide. Okay, monosaccharide ही आपके absorb होंगे, otherwise disaccharide आपके absorb intestine से नहीं होंगे. So these monosaccharides they are transported against the concentration gradient. Against the concentration gradient means from a low glucose concentration to a higher concentration. Let's say this is the glucose transporter. Okay, this is the glucose transporter. This is sodium dependent glucose transporters. And what it is doing, on the one end, it binds to the sodium, and on the another end, it binds to the glucose. It binds to the glucose and sodium at separate sites and then transports them through the plasma membrane, okay, of the intestinal cells. So this sodium is transported from a region of higher concentration to lower concentration, and at the same time glucose is transported against the concentration gradient. One thing. Secondly, the carbohydrates, they are also transported by the facilitative transport. There are two carbohydrates, fructose and mannose, that are transported across this brush border epithelium. You can see there is finger-like projections, the microvilli of the intestine, and this happens by facilitative diffusion. Okay, this happens by facilitative diffusions, and this facilitative diffusion also requires certain kind of transporters which are known as glucose transporters 5, GLUT4. They require glucose transporter 5. Moreover, the movement of glucose in the facilitative transport is from the region of higher concentration to the region of lower concentration. Moreover, there are certain sodium independent transporters as well. Sodium independent transporters, such as we say it's the GLUT2. This is also helping in transporting the sugars out of the mucosal cell and entering the portal circulation. Portal circulation. Now, when the carbohydrates, the monosaccharides, they enter into the circulation. From there, from the blood, now they will enter into the cell. Why? Because all the metabolism is going to be happen in the cell. All the reactions of the catabolism and anabolism, they are now going to be happen in cell. So let's now see how the glucose enters the cell. So, when we see that the glucose concentration is very low in the cells as compared to the blood, yeah, let's say plasma, and for human, it's less than 100 mg per dl. So glucose is not going to enter into the cell by simple diffusion. No, it never enters into the cell by simple diffusion. So how it enters? How does it enter? So there are transport systems. There are two specific transport systems. by which the glucose will enter into the cell. One system is insulin independent transport system. This is carrier mediated uptake and which is not dependent on insulin. Okay, insulin has no role to play in this. Okay, this is operative in hepatocytes. We have to notice down the location: hepatocytes, erythrocytes, and brain. See, now remember, now one thing is there, that RBCs needs energy, instant energy, because they have to constantly transport the oxygen uh, towards the cell and take away carbon dioxides away from the cell. So they cannot wait for any insulin to come to act upon the membrane of the cell. Hence, there the transport system is not dependent on insulin. Similarly, happens with the brain. So insulin plays no role whenever there is the transportation of glucose into the hepatocytes, erythrocytes, and brain. Simply, there is a carrier, and the carrier mediated transport takes place. Now, let's see the insulin dependent transport system. It specifically occurs in muscles and adipose tissue. So there are certain glucose transporters involved in this insulin dependent transport system as well. They may range from GLUT1 to GLUT5. GLUT is nothing but GLU for glucose and T for transporters. So there are various types of glucose transporters, and you know why they are named differently, because they are present in different cells. GLUT1 is somewhere else and GLUT5 is somewhere else, and they exhibit, we can say they exhibit tissue specificity. Okay, now let's say GLUT1 is found in erythrocytes, whereas GLUT4 is abundant in skeletal muscles and adipose tissue. See, remember, when we are talking about glucose transporters, we are also talking about glucose transporters in this slide, that where there is insulin independent transport system. साथ में ही दे दिया है ये. RBCs में कौन सा होता है? GLUT1. Although the transport system in RBCs, it is non-insulin dependent. Since the information is about glucose transporter, so it is mentioned here. Okay, so there is GLUT4 in skeletal muscles and adipose tissue. And whenever a person is suffering from type 2 diabetes mellitus, we see that there is insulin resistance in such patients. Okay, so why? Because there is a reduction in the quantity of GLUT4 in insulin deficiency. We will be covering this topic when we will cover the diabetes in detail. Okay, so let's see the diagram, that let's say that blood glucose levels are normal. Blood glucose levels are normal after eating and after digestion, after absorption. जो आपका ग्लूकोज है वो ब्लड में आ गया है. Once your glucose comes into blood, signal goes to the hypothalamus. Signal goes to the hypothalamus. The hypothalamus again signals the pituitary. Okay, blood glucose जैसे ही blood में आया, signal goes to the hypothalamus. Now the hypothalamus, which signals the pituitary. Pituitary is the master gland and hypothalamus is the master of master gland. Now then pituitary says to pancreas कि ब्लड में ग्लूकोज आ गया है, दरवाजे पर कोई आया है, उसकी खातिरदारी का इंतजाम किया जाए. ठीक है, सो पेंक्रियाज ने खातिरदारी के लिए क्या किया? पेंक्रियाज ने बनाना शुरू कर दिया इंसुलिन. यू नो, पेंक्रियाज में आइलेट्स ऑफ लैंगरहैंस होती हैं, बीटा सेल्स होती हैं जो इंसुलिन बनाती है. ओके, सो नाउ दिस इज अ टिपिकल मसल सेल. और हम इस मसल सेल के पास क्या देखते हैं? दैट इट हैज, यू सी, इट हैज receptors for insulin. Here are the receptors for insulin. So जैसे ही ब्लड में ग्लूकोज आया और पेंक्रियाज ने इंसुलिन सिक्रेट की, this insulin, it binds to the receptors. It binds to the receptors and signals the glucose transporters. these are the glucose transporters. This insulin is going to give signal to the glucose transporters, और सिग्नल देने के बाद फिर वही बात होगी कि दरवाजे पे कोई आया है. अब ये जो ग्लूकोज ट्रांसपोर्टर्स हैं, ये एक वेसिकल के साथ अटैच्ड होते हैं. दे आर attached to a vesicle. Okay, and when they receive a signal that glucose is there, the vesicle fuses with the plasma membrane. The vesicle fuses with the plasma membrane, and here you can see that the glucose transporter, okay, the glucose transporter is dragging in the glucose. The glucose transporter is dragging in the glucose. Okay. अब जब ये एक बार ग्लूकोज को अंदर लेके आ जाएगा, सेल के अंदर लेके आ जाएगा ग्लूकोज को, ठीक है, then what will happen? This glucose will be immediately phosphorylated, and these transporters, all these transporters will again, be again come down to so as to charge themselves, okay, so as to charge themselves while they will have to transport the glucose next time. Okay, so puri ka pura mechanism, it is based on the insulin. So when we see such kind of mechanism, they require lots and lots of efforts. Okay, इसमें effort लगता है, and yes, they are being operated. And once the glucose enters the cell, so that it do not goes out along with the concentration gradient, it is phosphorylated. It is phosphorylated. The phosphorylation is done immediately. So that's all for today. So see you tomorrow. So next lecture will be about the glycolysis. Thank you so much. In case of any queries, you can comment and you can always ask your queries. Thank you so much.